Assalamualaikum warahmatullahi wabarakatuh. Hello everyone, my name is Putri Ningsih. I am a medical student of Putri University. So, in this opportunity, I will explain to all of you about the journal. The title of the journal is Beta-endorphin response to aerobic and anaerobic exercises in restart milk breaths. This journal was written by Rustika Flora, Muhammad Sukarnoy, and Sukirno. I got this journal from Medical Journal of Indonesia. In this presentation, I will explain to all of you about background, aims, method, result, and conclusions of the journal. First, I will explain about background and aims. So, what is beta-endorphin? Beta-endorphin is released from the anterior pituitary gland in response to psychological conditions and somatic stress. Athlete psychological stress prior to competition trigger beta-endorphin release. During physical exercise, psychological and physical stress synergistically increase beta-endorphin that plays a primary role as an analgesic. Beta-endorphins also inhibits the transmission of phencyl and induces euphoria. This journal uses rats to become their sample. Rats have been used to answer many hypotheses in exercise physiology, including physical performance, because they adequately mimic human responses to exercise in terms of blood biochemical examinations. The physiological responses, resemblance in rat and human blood after exercise, indicates that the research involving rats for the exercise physiology is justified. Many studies have been conducted to investigate the correlation between physical exercise and endorphin release and their correlations to analgesic effect and euphoria. Clinical studies have also been done to determine the level of endorphin before, during, and after physical exercise, though the results are still debatable. This inconsistency occurs due to different inferences for defining types of physical exercise. The study aimed to compare the level of beta-endorphin in brain tissues stemming from aerobic and anaerobic physical exercise. Next, I will explain about the methods. This current study was conducted in Bioscience Research Laboratory, Palembang in April 2017. The methods, I will explain to all of you about subject, procedure, provision of homogeneous and determinations of endorphin levels. First, I will explain about the subjects. So the subject of this study was 35 Pisarmel rats with an age 6 until 8 weeks old, weighing 80 until 100 grams divided randomly into 7 groups. Such as control group, there is no physical exercise, 3 groups with aerobic physical exercise, and 3 groups with anaerobic physical exercise. The subjects were treated with physical exercises on a treatment for 6 weeks. Since the research was conducted for 6 weeks to avoid dropout samples such as leg injury, electric shocks, and rats getting too lazy to run on the treadmill, they added 2 more rats to each aerobic and anaerobic group. So the total sample was 49 rats. But during experiments, there were 9 rats that experienced leg injury and 5 rats that suffered electric shock. So for the data analysis, they analyzed 35 samples, 5 samples for each 7 group that didn't experience leg injury and electric shock. Because of the habit of the rat to be more active at night, physical exercise activity were carried out at night. Before the treatment, the animals were acclimated for 2 weeks to adapt them to research environment treadmill. Now I will explain to all of you about the procedures. So the procedure of the physical exercise treatment were adapted from a study by Flora et al. Physical exercise was conducted by placing the rats on animal treadmill, running at speed of 20 meters per minute for 30 minutes for aerobic physical exercise, and for anaerobic physical exercise, running at speed of 35 meters per minute for 20 minutes with 1 minute interval of every 5 minutes. Before and after running on a treadmill, the body weight of the animals were taken. In the 6 weeks immediately after physical exercises, the rats were decapitated and the brain tissues were collected from all groups. Now, I will explain to all of you about preparation of homogenate and determination of endorphin level. After brain tissues were collected, the homogenate was prepared using a homogenizer. The procedure was adapted from a study by Flora et al. The level of endorphin in brain tissue was determined using rat beta-endorphin ELISA kits. Now I will explain to all of you about data analysis. The data were represented in mean and standard deviations. Differences of mean endorphin level between control and treatment groups were analyzed with independent t-tests. Meanwhile, differences of mean endorphin levels between the control groups, aerobic groups, and anaerobic groups were analyzed with one-way analysis of variance. Next, I will explain to all of you about results. So from the table 1, we can see after the 6-week treatment, it was found that the mean body weights of increased in all groups. However, the increased weight of the control group were found to be higher than those given aerobic and anaerobic physical exercise. We can see too in figure 1, the brain tissue beta-endorphin level in rats treated with physical exercise revealed that both beta-endorphin level in aerobic and anaerobic group were higher than the control groups. In subjects having aerobic exercise, the greatest increase was found in the 1 times per week exercise. Meanwhile, in anaerobic physical exercise, the highest increase was found in subjects which had exercised 7 times per week. So, from this present study, both aerobic and anaerobic physical exercise can increase the level of beta-endorphin in brain tissue of lizard rats. But there are some limitations of this study, such as not all rats are capable of finishing 6-week program of physical activity, especially when performed 7 times per week. Injuries to legs and tail, age, and body weight influence their abilities to run on the treadmill. Also, some rats tend to be lazier or reluctant to perform physical activities as they age. So, a large number of rats were needed to replace dropout subjects and to repeat the study protocols. Consequently, the finishing times of each group were different. In conclusion, physical exercise, both aerobic and anaerobic, can increase the level of beta-endorphin in brain tissues of male wizard rats. The intensity of physical activity affects the release of beta-endorphin. So that's all about my presentation. Thank you for listening. I hope you have a nice day. Bye bye.